Bruce Beutler was young when he knew he wanted to be a scientist.
I thought how interesting that molecules can assemble themselves to be a dog or a cat or a person, something more than the sum of their parts. That was when I was 8 or 9. When I was about 13 or 14, I started to work in my father's laboratory. I worked on red cell glutathione peroxidase, and we actually published a couple of papers together.
His father, Ernest Beutler, was a prominent hematologist and department head at the Scripps Research Institute in La Jolla, California. Bruce finished high school at 16 and university at 18. He left to study medicine, but returned to research in Southern California as the head of genetics at Scripps 11 years ago. In the mid-1980s, Beutler isolated tumor necrosis factor, TNF, in mice. It's produced by cells involved in immune response.
Many forms of inflammation are caused by this molecule, TNF, including rheumatoid arthritis and Crohn's disease, a common kind of colitis. And now inhibition of TNF in those diseases and a few others is really the mainstay of therapy.
Beutler also worked on drugs to block TNF.
One of them is now marketed under the name Enbrel.
I had some intense pain. It progressively got worse. My rheumatologist told me about Enbrel.
From the perspective of the discovery of TNF and how to block this molecule, millions of people are millions throughout the world.
A lot of scientists are focused on a specific problem, and they attack it in what's called a hypothesis-driven manner. Bruce's research is really discovery. Let the animal, let the important physiology tell us.
Beutler started researching innate immunity in the mid-1980s, also hunting for what turned out to be the toll-like receptor and what would directly activate it. Beutler suspected it was lipopolysaccharide, LPS. It's the outer layer of gram-negative bacteria. After 8 years of false starts,
and it just so happened that there were out there in the world certain mice that didn't respond to LPS at all. We reasoned that these mice must have a mutation, probably in a single gene.
Finding the mutation took another 5 years.
The first step is to determine just which chromosome the mutation is on using a process called genetic mapping. Once that's accomplished, we can find even which part of the chromosome the mutation lies on, and all of the chromosome represents a long string of DNA bases held together as a single molecule and wrapped tightly around proteins called histones, as shown here. The question then is, which of the bases of DNA is defective? Exactly which gene is hit by the mutation to cause a difference between LPS responsiveness and unresponsiveness? It's just one base pair out of 2.6 billion that we're after. The consequence of that was to change a certain protein, TLR4, so that it no longer signaled the presence of endotoxin, of LPS.
He did do that at the time that we knew nothing about the mouse genome.
Or the human genome, or any large genome. We're doing our own sequencing.
The Beutler lab now has some 5,000 cages of mice and modern gene sequencing methods.
I'm sure the day will come when we see a mutant mouse in the morning and by evening we know what mutation caused the mutant phenotype.
What took him 5 years to do in the mid-90s now takes us a matter of months.
Postdoc Owen Siggs is studying white blood cells.
We make random mutations in mice, and then we take blood cells from these mice and look at them one by one to look for mice which don't have white blood cells. And the discoveries we make in mice are then applicable to humans and to patients with white blood cell diseases.
Bruce's lab bred mutations into mice for R. Theophilopoulos, who researches the autoimmune disease lupus. The mice didn't get lupus because 3 of their toll-like receptors didn't work.
That told us that in order to develop lupus, you need signaling by TLR3, 7, and 9.
Bruce plans to find all the genes that might be involved in immunity.
mutate every gene the mouse has in its genome and check mice with those mutations to see if they behave differently in terms of their immune function.
Bruce grew up in Southern California in a family of 4 kids, 3 of whom became doctors, including brother Steve.
We're all within 90 miles of each other and see each other frequently, several times a month if possible, and we take hikes like this whenever we can. I don't think there's any jealousy in the family at all. We're happy, amazed, proud of everything that he's done.
And they're preparing goodbyes.
In Dallas, I hope to establish a center for the genetics of host defense and to assemble a faculty with interests very similar to mine.
He's returning to the University of Texas Southwestern Medical Center, where he has been twice before.
I have to admit, I love California. I will miss Southern California.
Bruce was married in Dallas and had 3 boys before divorcing.
Until I was 15, maybe 16, my father and I both lived in Dallas.
Elliot is the second son. He lives in Seattle. None of the boys has pursued science.
I think that my dad always hoped that I would grow into those shoes, that I would eventually go into some field of scientific research.
Bruce's long-term, long-distance relationship with partner Nadia, the dermatologist, will apparently not face any new stress due to the move.
I live on the East Coast, and obviously he's on the West Coast, soon to be closer in Texas, but because of technology now, I feel connected to him. The internet has changed everything, emailing, talking all the time.
From my early days on, I really loved insects. It's a fantastic group.
Jules Hoffmann left his native Luxembourg after high school to major in biology at the University of Strasbourg in France.
There was a very strong department of zoology, and in particular there was a laboratory working on insect models.
Grasshoppers.
You would do transplantation of endocrine organs or tissues from one insect to the next.
Hoffmann's mentor noticed
there was never an infection appearing, so he concluded there must be strong antimicrobial processes in the insect system.
In 1969, Hoffmann took his PhD on that subject, continuing the work after joining the French National Research Agency, rising to become laboratory director. Under his watch, grasshoppers were replaced with the genetically tractable fruit fly, Drosophila.
This is really the basic experiment, pricking the insect with a needle which has been dipped into bacterial fungal cultures.
After years of biochemical and cloning studies of tens of thousands of fly lines,
we had identified that the insect was producing antimicrobial peptides to fight off invading microorganisms.
the scientists then studied how the genes encoding these peptides are induced in immunity response. They found two distinct pathways, the IMD and the toll.
So in all, a fly, to be able to withstand infections by various microorganisms, needs the two pathways.
Mutant flies with no IMD would succumb to gram-negative bacterial infections, such as E. coli. Toll-deficient flies die of fungi and gram-positive bacteria.
By introducing in this fly a normal toll receptor, and the fly again will be able to withstand the infection as wild types. This was really a very early evolutionary response to infection. It attracted considerable interest in the community because it was the basis of a molecular analysis of innate immune defenses.
As opposed to adaptive immune defenses found only in vertebrates which produce antibodies. After some 30 years of studies, a paper was published in 1996.
The studies which have been developed over the last 15 years or so have shown that the innate immune response activates adaptive immunity and can orient the type of adaptive immunity.
It really changed the way we see diseases. There are many biotechs that are working on these receptors, using them to find new vaccines, to challenge in other ways the immune system, even in cancer.
If they were to escape, you would be in bad shape.
Hoffman's innate immunity findings led to lab mate Dr. Elena Levashina's studies of mosquitoes.
What makes mosquitoes vectors of malaria? What are the genes which make them susceptible to malaria? And eventually how we can modify expression of these genes so that mosquitoes can be transmitting it younger. Jules is an incredible scientist. He is very enthusiastic about everything, and he really likes to bring new aspects into research.
He has a very good feeling of these scientific programs. He can give some very good suggestions and many good questions.
Hoffman's protégés are working on antiviral defenses in flies.
Viral infection is a very serious threat for the human disease. The Drosophila can offer kind of fundamental molecular basis for the viral infections.
I'm trying to establish some of the biochemical approach so that we can know that how this immune response is kind of evoked and then prolonged and maintained.
What I find really amazing is the dedication and persistence of Jules Hoffman working on insects. Big organizations or university presidents wanted him to work on humans, immunology. Oh, that's more important than the immunology of insects. But he continued.
A potent stimulus was his father, Jos Hoffman, a high school biology teacher who also drew and studied insects.
My father introduced me to science. I started at the age of 13 going out into nature with my father and collecting insects and keeping them in jars and identifying them, of course, under the microscope.
Jules met his Strasbourg-born wife, Danielle, at the university, where she also majored in biology and worked in the same lab.
I remember a handsome young man who one day picked me up to go to a party. He volunteered. And,
why did you do that?
Just imagine why. Well, I was, at the time I was looking for a partner and she was very good looking.
They began dating.
Of course, it was easy. I mean, we were in the lab the whole time together.
They married 2 years later. After Danielle got her PhD, she eventually joined Jules' lab as an administrator.
We were at a given moment working both on steroid hormones in insects and on immunity. And I was always wondering, this is not one topic too many. And we discussed it together, and Danielle was adamant that immunity would be more interesting on the long run.
A decision that helped them win many prizes.
I was very happy, and I think I was supportive.
After the couple's 2 children were born, Danielle went to Sweden for postdoctoral work.
Jules was very supportive because he took care of them for that time.
Son, Mark, has a doctorate in mathematics, and daughter Isabel, a PhD in microbiology. 4 PhDs in the family.
Everything is in the genes. Certainly it was the atmosphere of the family. We were very close.
Danielle retired early.
I was just too tired to, and I had also interest in music.
Jules aims to stop working at 75 in 8 years' time.
We hike a lot, we're very active. The important thing is to stay strong in good health until then, you see. I do this every evening. French paradox is that to have a good health, you have to have red wine every day, a little bit.
Jules has about 2,000 bottles here.
Those are the great Bordeaux. The 97 Chateau Palmer, which is a Margaux wine.
The whites and
Chateau Yquem 91. I've always been very much interested in history and in languages, the evolution of mankind, humanitarian aspects, and where do we go, why are we here, how the Christian Bible evolved over 2,000 years. This building dates to the 18th century.
As a life scientist, how does he see life?
I don't think that working on animals gives you any better insight into what life is all about. Science is a way of learning modesty. To such big philosophical questions, I don't think that science brings you any nearer. Otherwise, we would really be privileged people. How would I like to be remembered? As a nice person who tried to do what he could, and I did my best. I know we have billions of people in that situation over the world, so probably I will not be remembered. Is that a sad conclusion?
The Medvedov Immunobiology Lab at Yale University.
There may be very rare cell populations and you just don't see it.
Throughout the lab are students and postdocs doing research that is outside of immunology.
Right now I'm working on why a seemingly homogeneous population of cells choose to respond differently to a stimulus.
By working in different fields and having really talented people in my lab who work in those fields, it allows me to expand even though I cannot be an expert in all of these fields.
If it's interesting, he's willing to go along with it.
This was the way Ruslan Medvedov's mentor, Charles Janeway, thought.
He actually had this ability to have this grand unifying theories and synthesis of multiple different fields.
Janeway had a theory on innate immunity published in a 1989 paper. It said,
there should be a distinct form of immune recognition that would detect certain features of microbes that are common to all microbes, and that would allow our immune system to tell the difference between microbial and non-microbial.
Janeway's hypothesis was so attractive to Medvedov that as a biochemistry PhD student in Moscow, he started communicating with Janeway by email. He then parlayed a fellowship at UC San Diego into a postdoctoral position in the Janeway lab at Yale in 1994.
If I couldn't get to his lab, I think I probably wouldn't go into immunology at all.
When Medvedov started investigating innate immunity in vertebrates, little was known about the way it worked.
It was thought that innate immune system simply provides the first line of defense during an infection to maintain microbes enough until the real immune response is activated, which is adaptive immune response.
Medvedov wanted to know how the antigen-presenting cell, the APC, knows that a pathogen is present so that it sends signals to T cells and activates an adaptive immune response.
What I was looking for initially was a receptor that would be able to activate a certain signaling pathway.
The receptor he found was similar to the toll receptor in Drosophila flies.
What we call homologous, meaning it's human version of the Drosophila gene.
That gene had only been associated with embryonic development in flies. Just months later, Jules Hoffman published his work showing toll was also involved with innate immunity.
That's when everything became sort of fall into places.
Medvedov used an artificial method to activate the receptor he had found in humans and show it can induce an adaptive immune response. These receptors became known as toll-like receptors, or TLRs. The one Medvedov identified was TLR4. About a dozen have now been discovered in mammalian species.
There was an explosive growth and research on these receptors. Some of them are involved in recognition of bacterial pathogens, others viral pathogens.
Down the hall from the Ruslan Medvedov lab at Yale is an admiring colleague in the lab next door.
I've never met anyone that kind of compares to Ruslan in terms of the way he is able to synthesize his thoughts into very cohesive programs. And that's what he translates in his lab.
Professor Akiko Iwasaki has her own immunobiology lab. She married Ruslan Medvedov 4 years ago. Although they work next door to each other, they say there's never enough time together.
Because we have own family in the lab, meaning that I have many people that I consider very important to mentor and foster their scientific career. And he also has more than 20 people in his lab that he's in charge of.
Home is half an hour outside of New Haven, in a new house big enough for visiting relatives.
We had, like 12 people here for 2 weeks with Akiko's extended family. So for that, we needed every single room in the house.
Their daughters are Emmy and Naomi.
I'm very pleasantly surprised how wonderful he is as a father.
Their parents speak Russian and Japanese to them.
First time we heard her speak English was when somebody came, and some guests were here, and she completely fluently spoke English with them.
He's gotten a lot more tolerant, which you have to be as a parent.
So many things that worried or bothered or annoyed me or excited me before now don't seem as important.
When Ruslan was at school in Moscow, the Soviet Union was coming apart. There was no money for experimental science.
I spent most of my time basically in libraries reading. And I already had sort of interest in multiple areas of biology, and I was just reading whatever I could find on various areas related to molecular evolution in particular.
He's constantly reading and trying to understand different things and new fields.
That's my biggest frustration, that I wish I had a couple of parallel lives to do reading, the other readings I wanted to do.
Something that makes him unique and special is his really broad interest and his ability to connect things that I think a lot of people wouldn't necessarily connect.
Rachel Rosenstein is studying allergy, but learns more.
We do something called Molecule of the Day, where weekly we have a session where someone presents on a molecule.
Molecules that have some interesting, important functions, but completely outside of our field.
And somehow this molecule then ends up connected to ideas related to people's projects.
And those projects lead to discoveries like how innate immunity works. One of the things it does is respond to pathogens with inflammation. And more, inflammation is being connected to modern diseases like adult-onset diabetes, cancer, and neurodegenerative diseases.
There's a lot of fundamental unknowns about how that inflammation is initiated, what maintains it, and how it contributes to disease exactly. That, I think, is the next 10 years is going to be a lot of very exciting and important findings in that area.